Bismillahirrahmanirrahim. In the name of Allah, the most merciful and the most beneficent. Countless salutation on Hazrat Muhammad sallallahu alaihi wasallam, the last prophet. So, in last lectures we were discussed about computational drug designing and conventional drug designing, limitations and benefits of computational drug designing and conventional drug designing. Then we also have a look to the preface and foreword of the book Quick Guideline for Computational Drug Design. So today we will study about the introduction of structural bioinformatics in chapter 1 of the book Quick Guideline for Computational Drug Design. So there are so many branches of bioinformatics, as every field or domain usually have. So in this book we will focus on structural bioinformatics, like we have structure biology in the domain of biology, in which we just study about the structures of biology. So we did not have much participants yet, so we will wait for 1 minute for the participants to join. Sabika Zulfikar 16823.
Assalamualaikum, sir.
Walaikum Salam. We don't have participants today. We have only two participants. Everything is okay?
I don't know, sir.
Okay. So we were discussing about introduction of structural bioinformatics, chapter first of Quick Guideline for Computational Drug Design. The abstract of this chapter is bioinformatics is an emerging modern biology to solve the biological problems with the help of computational approaches by utilizing mathematical and statistical techniques. There are many databases of protein and gene sequences. The protein sequence can be retrieved from UniProt, and three-dimensional structure prediction can be performed by utilizing homology modeling, threading, and ab initio. The predicted structures can be evaluated by utilizing numerous model evaluation tools. So, first of all, in structural bioinformatics, if you want to predict the structure of any compound or biomolecule like DNA, RNA, protein, obviously you have the structure of DNA and RNA, you have to predict the structure of proteins, or you have to predict the interactions of protein-protein interaction, protein DNA interaction, or protein ligand interaction. So the selection of protein is very important, or the selection of specific molecule. So you should be very careful for the selection of the target protein. You should know about the target protein. Your literature should be enough on that specific protein. You should know about the protein, the mechanism of the protein, the behavior of the protein, the amino number of amino acid of the protein. Is it polar amino acids? How many polar amino acids are there? How many non-polar amino acids are there? Which region should be hydrophilic? Which region should be hydrophobic? And what will be the specific nature of the protein, how this protein acts, how this protein behaves, how this when this protein becomes functional, how many signal peptides, how many amino acids and signal peptides this protein has, in which mechanism this protein is involved, the mutation of this protein leads to which disease, or how many mutations it has. It has sense mutation, nonsense mutation, frame shift mutation, transvariant mutation, transition mutation, chromosomal mutation, which mutation it has. So for the selection of target protein, you have to know all these information, or you have to retrieve all this information for better analysis. Then you also have to know that which from which family it belongs, how many members this protein has in his family, and how many structures are available in its family, what is the function of this family, how specifically this family prevails, and how many what will be the evolutionary history of this family, and is there any specific prosthetic group for this family, or for this target protein, or cofactor for the function of that specific protein. So everything you should know about the protein. You should work hard to gather all these information, and then you will decide to do further experiments on that specific target protein. So the selection of experiments and the selection of uh, project is very important. It should be in right direction. It should be in your expertise. You should know much about the literature. So all these points are very important for that. So we were discussing about structural bioinformatics, in which structural domain of the bioinformatics will be discussed, the structure prediction of protein, how to predict the structures, which approaches will use, like homology modeling, threading, and ab initio. As I remember, we already studied about these approaches of structural approaches, that is homology modeling, also known as comparative modeling, threading, that is also known as fold recognition, and ab initio. After the structure prediction, three-dimensional structure prediction, so there will be the evaluation of that predicted structure. So there are numerous tools are available for the prediction of for the evaluation of predicted three-dimensional structures. So every tool has its own pros and cons. So selection of specific tool is very important for the experiment. Then we have a motivation for that specific work, and anyone wants to read it, please raise your hand. Anyone wants to read, please raise your hand. Samra 168448. जी बेटा, please read it. Samra 16848. Asma Khalil 16814. जी बेटा,
Yes,
please
sir.
read it.
Motivation. The purpose of this chapter is to introduce reader to the basic views about the bioinformatics and some basic concepts about bioinformatics in computer-aided drug designing. The basic measure approaches and the availability of tools and servers will also be explored in this chapter. The foremost step of most of the computational analysis is the acquiring of sequence and alignment of the protein of interest. This chapter will also sketch the portrait of the complete book.
Next.
On 5th of October 1981, an article titled Next Industrial Revolution Designing Drugs by Computer at Merck was published in the Fortune magazine. It is considered as the step on computer-aided drug design. Till now, many developments have been done in computer-aided drug design and the high throughput screening. Techniques also
High
help to
throughput
develop
बेटा,
the novel.
high throughput नहीं, high throughput screening.
Yes, high throughput screening technique also have done in computer-aided drug design, and the high throughput screening techniques also help to develop the novel therapeutic targets. This techniques develops on the automation
Depends on the
for the
automation
screening
now.
depends on the automation for the screening of millions of molecules to find biologically active compound. The advantages of this technique is to screen large libraries and scrutinize
Scrutinize.
the scrutinize the biologically active molecules. The HTC techniques usually
HTS
result in the
technique.
HTS techniques usually result in numerous hits, and some have the capability of becoming lead molecule despite the hits rate below low.
Being
From
low.
the last decade, rate being low. From the last decade, number of biologically active compounds have been reported by applying the computer-aided drug design. This approach reduces the workload and the time phase.
Thank you. So in this, there will be motivation that what and how and when the computer-aided drug design starts, and the more specific technique is high throughput screening that we already discussed in our previous lectures, that in conventional throughput screening, we usually researchers needs years and years and number of expertise, and also they should need much more money to scrutinize a specific compound for a specific protein or a specific disease. So if the researcher has almost 1 million compounds from any library, he will spend almost 10 years or maybe more than 10 years to scrutinize all the compounds specifically for a specific target by utilizing the experimental approaches. However, when virtual high throughput screening just invented, or computational high throughput screening, it just reduces the time frame and give a good sketch for that specific library of 1 million compounds in maybe 1 year or in 6 months. Now researchers are very much experts in this domain and also have resources and big servers, powerful servers that just analyze 1 million compounds in 1 year, or maybe sometimes more, less than 1 year. So they just perform all the necessary experiments. They just perform all the necessary experiments computationally and give a sketch that suppose these 200 or these 500 compounds or these 100 compounds are much more potent or effective for that specific protein or disease by using computational analysis. Oh, after that, an experimental biologist or wet lab expert can analyze those 100 compounds or 200 compounds in 2 to 3 years, and we can have a good sketch of that compound against that specific target protein or disease. By the computational drug designing and high throughput virtual screening, and the time frame just reduces down. So now researchers first just analyze the millions of compounds and the libraries of millions of compounds through computational high throughput screening or computational drug designing approaches to narrow down the window of those million of compounds into hundreds or tens of compounds. After that, those compounds will use for further experiments for further experiments. So if the compounds are mutagenic or maybe carcinogenic or not fulfilling the requirement of drug likeness or not fulfilling the requirement of Lipinski rule of 5 or a rule of 3. So the researcher just filter out those compounds through computational approaches, or it will take maybe 1 month to scrutinize all those compounds computationally, millions of all those compounds that are mutagenic or carcinogenic. Now there are many tools available that just help us to analyze the properties of compounds that are mutagenic or carcinogenic, and also many databases are available in which the specific compounds or the libraries that has those compounds, and the properties of each compounds are mentioned in the databases, or numerous tools are available that you can use to analyze that these compounds are mutagenic or carcinogenic, and or any other these type of limitation the compound has. So you don't need to use those compounds for your experimental work specifically. However, sometimes those compounds have the potential, moreover, but according to theoretical biology or theoretical means, those compounds should be filtered out to save the resources, as many of, or maybe 90% or 80% of compounds, those are carcinogenic, we don't know, we don't use. In carcinogenic case, maybe 100% or 99% compounds we filtered out. Similarly, in mutagenic cases, however, we can make sacrifice on Lipinski rule of 5 or rule of 3 or drug likeness properties. For that, sometimes we just use those compounds for experimental means. However, the carcinogenic and mutagenic compounds we don't use for the specific use for other experiments. As you know, carcinogenic compounds are very dangerous if we are going to cure or treat a specific target protein or disease, and then we also gift a mutation or a cancer to that patient, so it is not a beneficial deal. So that's why these compounds didn't use or specifically filter out for further experiments. So who is going to read the definition of bioinformatics? Please raise your hand. I suggest you guys to read more and more. I observe that the reading or the confidence of reading is not much in among you, all of you. You guys hesitated or have many pronunciation problems. So please read as more as you can. At least read 1 or 2 articles in a week specifically related to your domain, and try to learn and observe from. Okay, now we have a guy or a girl, maybe.
Assalamualaikum, sir.
Walaikum salam ji, beta, please, from bioinformatics.
Bioinformatics. Bioinformatics is an interdisciplinary field that develops methods and tools for the understanding of biological data. As it is an interdisciplinary field of science, so the techniques of computer sciences, statistics, mathematics, and engineering are used to process the biological data. Bioinformatics is an emerging scientific field that utilizes computational, mathematical, and statistical approaches for in silico solutions of biological problems. Here, in the beginning of this book, we want to explain that the bioinformatics is a vast field and have uncountable tools, software, servers, database, and also various approaches and techniques to perform the same task. We try to describe the highly cited and effective approaches in this book and also try to elaborate in a simple manner for the understanding of the layman and the beginner.
For the layman
Bioinformatics
and the
approach.
beginner.
Layman and the beginner. Bioinformatics approaches. Bioinformatics tool including softwares for mapping, analyzing, and aligning the DNA and protein sequences for analysis, creating and viewing the three-dimensional 3D models of protein structures according to the basic approaches of bioinformatics. There are two fundamental ways of modeling biological systems, like static and dynamic. Static, it includes the sequences of proteins, nucleic acids, and peptides. It also contains the interaction data for static entities including microarray data, networks of proteins, and metabolites. Dynamic, it includes the structures of nucleic acids, ligands including metabolites and drugs, and peptides. The structures studied by using the bioinformatics tools are not considered static anymore. Systems biology also comes under this category, including reaction fluxes and variable concentrations of metabolites. The multi-agent based modeling approaches capturing cellular events such as signaling, transcription, and reaction dynamics are also considered as the dynamic approach of bioinformatics. There are numerous subfields of bioinformatics, but in this effort, the main focus will be on structural bioinformatics. Structural
Thank you.
bioinformatics.
Thank you,
Yes,
Tahmina.
sir.
So, in this, first of all, we should know what is bioinformatics. Usually, students have a very simple definition that I heard, and it's not a proper definition. Like when I ask from the students, you more than 70% students told me that marriage between computer science and biology is bioinformatics. It's not the proper definition. I don't know from where you guys got this definition. So in actual, the main concept of bioinformatics was to use the statistical mathematical approaches, algorithms, ideas, techniques to solve the biological problems with the help of computation power. So bioinformatics is specifically an interdisciplinary field that just utilize the approaches, techniques, algorithms, methodologies, and of statistical methods and mathematical methods to solve the biological problems with the help of computational powers. So this was in actual the bioinformatics. Now it's a very vast domain, Every biologist almost needs a bioinformatician. So many involvement of bioinformatics in all the life sciences and biological sciences domains. Bioinformatics is the field of science at the cutting edge of two fastest and latest growing technologies, molecular biology and computer sciences, with the interference of statistical and mathematical models. So, bioinformatics helps the researchers to lower down the expenses and also to reduce the time. So there are numerous solutions available and there are so many domains of bioinformatics. However, in this book, only structural bioinformatics will be highlighted, as we are working on as structural bioinformatics just leads towards quick guideline for computational drug design. So for designing a computational drug, so first of all you should have the structure of that specific target protein. If it is not resolved by X-ray crystallographic technique or NMR, then you should have to predict the three-dimensional structure of that specific protein. As you guys know, the function of the protein depends on the structure of the protein. So we have Shan Aziz here. Do we have Shan Aziz here? Or anyone else? Please raise your hand and let me know. You guys studied about NMR and X-ray crystallographic techniques. Anyone please help me? You guys, Tehmina Latif, please raise your hand. Okay, who is this? आयशा मकबूल जी बेटा पढ़ चुके हैं हम पहले
No, sir.
एनएमआर एंड एक्सरे क्रिस्टलोग्राफी पढ़ चुके हैं एक्सरे क्रिस्टलोग्राफी जी बेटा आयशा मकबूल ओके आई थिंक शी सेड yes we studied. So then you have a sketch of working of X-ray crystallography and NMR, and if you don't attend the lecture, you can study about that from different sources to understand what an actual X-ray crystallography and NMR and how these approaches just resolve the three-dimensional structure of specific biomolecule. And if the protein structure is not available specifically with the help of X-ray crystallography or NMR, then we have to jump on computational approaches to solve the three-dimensional structure of that specific protein, that target protein. So after the structure of protein, we can perform further experiments. If we don't have the structure, we cannot perform the experiments for the understanding of binding domain, which residues are important, which residues are interacting residues, how these amino acids just interact with other amino acids, and what change will happen when there will be specific interaction of that interacting residues. As you know, when there is some interaction with other proteins or other biomolecules, the protein changes its structures because protein is always in dynamic form. So you should know about that if you will have the structure, and if the structure is not available and you don't have the structure, you cannot predict the three-dimensional structure specifically, then you cannot perform further experiments. So now we are going to move down on structural bioinformatics. Anyone wants to give a read, please raise your hand. Okay, please raise your hand and I will give you the chance, everyone. Please, Aisha Makbul.
Assalamu alaikum, sir.
Walaikum salam.
Structural bioinformatics protein structure prediction is an important application of bioinformatics. It is the prediction of protein structure from the amino acid sequence.
It helps to predict the folding, secondary, tertiary, and quaternary structures of the target protein from its primary structure. Structure prediction is fundamentally different from the inverse problem of protein design. The amino acid sequence of a protein's primary structure can be easily determined from the sequence of the gene that codes for its primary structure uniquely determines a structure in its native environment. The knowledge of primary structure is vital to understand the function of protein. Structure information is usually classified as one of the secondary, tertiary, and quaternary structures. Homeways, a,
A
vi-
viable
a,
general
vi-
solution.
a viable general solution to such prediction remains an open problem. Most of the efforts so far have been directed toward heuristics that help to solve these problems computationally.
Thank
In
you,
the
beta. Thank
in
you. Anyone else? When you raise your hand, please raise your please don't lower down your hand so I can know who is in the next queue for reading. There is a lady Aruj, and all the time she lowered down her hand and I could not give her the chance. Sorry for that. Please raise your hand. Okay, still raise your hand. If you when you get the chance, then you can lower it down. So now we have Aruj Zara. Please. After that, Tehmin.
Assalamualaikum, sir.
Walaikum salam. Ji, please, beta.
In the genomic branch of bioinformatics, homology study of distant relatives is used to predict the function of a gene. If the sequence of a gene whose function is
Gene
known
A,
is
beta.
homologous to gene A, whose function is known, is homologous to the sequence of gene B, whose function is unknown, one could infer that B may share A's function in the structure branch of bioinformatics. Homology is used to determine which part of a protein is important in structure formation and interaction with other proteins. In homology modeling approach, the structure and functional information is used to
Predict the protein structure.
predict the protein structure by using the structural information of homologous proteins.
Thank you, beta. So, in structural bioinformatics, as we already discussed, that different stages of protein structures are, as you guys already studied about that: primary structure, secondary structure, tertiary structure, quaternary structure, and quinary structures. So you have to predict these structures sequentially. First of all, you should have the primary structure, that is the linear amino acid sequence of the target protein, and then you have to predict the secondary and tertiary structures, and you have to further work on that. For those, you have different approaches like Golgi modeling and threading and ab initio. If the structure of a homologous protein of the target protein is available, so you will use that specific protein structure that is available as a template structure to predict the three-dimensional structure of the target protein. So that is very important, because the backbone of that specific protein family usually have similar angles and properties. So we will take that specific backbone for our for the protein, the target protein we are going to predict. So we have the limitation of time, so we will join in next lecture from here, software and tools, and continue. Thank you so much everyone for joining.